All right. So, we're finally past binary fission. We're finally past the basics of the cell cycle. And now we're moving on to mitosis. Mitosis is a type of asexual reproduction. But before we go on, I'm going to review. Mitosis is not a part of interphase. Interphase is everything else in the cell cycle minus mitosis and cytokinesis. Now I want you to notice that I actually separated mitosis and cytokinesis. Cytokinesis is not a part of mitosis. It's after mitosis. Usually people group it together because it happens so quickly after mitosis, but it is not exclusively a part of mitosis, just like interphase isn't a part of mitosis. Notice. So if I could actually make that little line right here, what cytokinesis is in relation to the time spent on everything else, that's about how much time it would take. Just that little sliver. Alright? So, moving on. So, before we really get into mitosis, it's good to kind of give you an overview of what the steps to mitosis are. So I wrote them out. And I know I wrote cytokinesis, but it's important because it happens right after. So mitosis consists of prophase, prometaphase, metaphase, anaphase, telophase or telophase, and cytokinesis. Cytokinesis, again, is not a part of mitosis, but a lot of people group it in for the learning exercise because it completes the whole dividing off of the two daughter cells. So, just for funsies, I actually made kind of like a little, what? Mom's making fun of me. So I actually made like a little memory trick for you so you can remember the order of the names. And it's, please promise me all teal crayons. Prophase, prometaphase, like promise. Meta, all teal crayons. Anaphase, telophase, like telophase. And crayons, cytokinesis. Okay? So go ahead and write that down. Pause it. Write that down. Okay, cool. Alright, so who does mitosis? Good question. Eukaryotes with linear DNA, and there are multiple origins of replication. So, if you know anything about the genetic structure, the genetic components of a prokaryotic cell versus a eukaryotic cell, most of the time, prokaryotic cells are a lot more simple than a eukaryotic cell. Which also means that eukaryotic cells typically contain more genetic material and therefore take longer to replicate, if that makes sense. So, how eukaryotic cells solve the problem is having multiple origins of replication, where several different genetic machinery can attach to these various ories all over the genetic material, and they'll kind of all divide and conquer this genetic material, so it all happens a lot faster, and then they'll just zip it up together and you'll have nice one long strand. And because it's not a circle, there is a beginning and an end. There is no terminus. There's no terminal site like in the prokaryotic cells. So there's one ori, or there's multiple ories, and they all get glued together at the end. Alright? By DNA ligase, if you had to know. But you don't need to know that. So, the first phase is interphase. This is not a part of mitosis, okay? But interphase is extremely important in the cell cycle. Why? It looks really boring. That picture I drew you is pretty accurate what interphase looks like. It is a circle within a circle. The big circle being your cell membrane, the little circle being the nucleolus, okay? Or the nucleus. You can't even see the nucleolus because I didn't leave room for it. So, it looks boring, but it's highly active. Why is it highly active? Think about what we said in the last video. We're getting all of our ducks in a row, we're synthesizing DNA, and we're getting it ready for mitosis. There's a lot of building, a lot of growing, a lot of preparation going on in interphase, but it looks boring. So when you're looking at slides on your test, you need to look for the most boring looking cell, and that's interphase, literally, okay? The first step, so once we're done with, G1, S, and G2, we finally move in into mitosis. The first phase of mitosis, which is prophase, or please in my little memory trick. Prophase. So if you look at this, this is a really bad picture, but what you're looking for is condensed chromosomes. So you're still going to have your nuclear envelope, which is this little circle in the middle here. That will still be visible. Sometimes it will be slightly lighter than like in an interphase, but it will still be there, and all of the chromosomes should be, you should start seeing little noodly figures inside that nuclear envelope. You'll also sometimes notice that these little boxes on the sides, the little itty-bitty confetti pieces, those are called centrosomes, and centrosomes produce spindles. These spindle fibers are what are responsible for moving the chromosomes around during mitosis, but we don't really need to worry about them now. All you need to know is that in prophase, these spindle fibers actually start forming and start building up. They're made out of microtubules, which you also don't really need to know a lot about, but they're made out of microtubules, and microtubules they can like build up and break down like Legos. So in this phase, they're actually building up. Alright, and then, like I said before, this envelope tends to disappear during prophase, but it doesn't necessarily mean it's completely gone yet, okay? That doesn't happen until the next phase, prometaphase, or please promise in my little memory trick. Alright, so here you can see that my nuclear envelope is pretty much gone. It's like little dotted line around there. If it's not fading, it's probably completely gone, so don't be too torn up if you can't find a nuclear envelope in your picture. And then these things called kinetochore microtubules, MTs is what I'll abbreviate it as, microtubules form. Kinetochore is essentially the little, you can't really see it, but you see each of those X's is a homologous pair of chromosomes. And where the X's bisect, that right there is the kinetochore. It's actually like a little piece of glue that holds the two chromosomes together. So that's really important to know, because that little piece of glue, that little piece of adhesive that's holding those two, the homologous pair together, that is actually what the spindle attaches to and pulls from, okay? So that's why the kinetochore microtubules actually attach to the center of these X's of the chromosomes, and you'll see that. All right. You'll also notice that the chromosomes are much more defined in this phase. Your teacher, the book that you guys have, provides really, really awesome pictures, and I really advise that you check out those pictures because they are the most clear pictures of each of the mitotic phases that you can find. My pictures don't really do it justice. All right, here's my favorite phase, metaphase. It is literally the easiest phase to identify. You cannot screw this up. All of the chromosomes are in a line. That's it. You don't really need to know much more else. And the thing is, if you do the experiment and actually watch mitotic division, which takes forever, by the way, this is the quickest portion. It literally happens for like a split second because once they line up, they go right into the next phase. So metaphase is just when the chromosomes line up. All right, anaphase is the next one. Anaphase is the actual process when they first pull apart and separate. When they start to separate, you're in anaphase. If they're still in a line, you're in metaphase. The moment they separate, that's anaphase. Okay? It's the motion. It's not really a set period or a set picture. It's actually like the action of pulling apart is anaphase. Okay? So what you can see is they're splitting apart. If I could show this like moving, it would be the actual action of them splitting. What's really, really important is that this phase uses ATP. Why does it use ATP? Because these microtubules actually have small proteins on them that actually walk on the microtubules. It's really cool. But it actually walks on these microtubules, and in order for them to walk on the microtubules, it needs ATP for every single, 1 molecule of ATP for every single step it takes. It's really quite incredible. But what you need to know is that as it walks along these microtubules, pulling the chromosomes behind it, the microtubules just kind of fall apart. Remember, they're those little Lego pieces called tubulin. And those little pieces of tubulin are disintegrating off as you like walk across. It like goes away. All right. So those are disintegrating as the as the chromosomes are being pulled towards each individual side of the cell in anaphase. Okay. So anaphase, you go to the other side. Okay. Telophase or telophase. Teal. Please promise me all teal crayons. Telophase is actually where you can start to see that cytokinesis is about to happen, and the nuclear envelope begins to form again, and the chromosomes begin to unravel. So these might, like even the way I drew this, these are kind of each of the chromatids kind of like chilling on their own. They might not even look as organized as a chromatid anymore. They might just be like that curly string of DNA again. It might be that jumble all over again. If they are still attached and there are two very visible nuclei, it is in telophase, okay? And the spindles are also disappeared. You know, there are no need for the spindles if they aren't moving anywhere, and they're already inside a nuclear envelope, so that's telophase. And then, my favorite, actually second favorite because I like metaphase, cytokinesis, which I made a racial slur for last time, so I just drew it this time. It just basically cuts the cell in two, and there you have two individual daughter cells. Alright. So really quick though, before I wrap this up, there is something really important to know about cytokinesis. So cytokinesis does happen in all cells that divide mitotically or by binary fission. It doesn't matter. Ew, there's something in my hood. But they do it a different way. So in animal cells, they go about it with a contractile ring. Now in binary fission, this is very similar, okay? Now the contractile ring is where there's like a drawstring around the center. You know, like when you have a balloon animal and you twist the balloon and it pinches it off into two little sausage links of a balloon? That's pretty much what the contractile ring does. Is it pinches it from the outside in, all right? And that's for any cell that has only a plasma membrane. Now, there may be some prokaryotes out there that have just a plasma membrane, and if they do, that's how they would go about pinching off for cytokinesis. However, most plant cells and most prokaryotes have a cellular wall. This is a very poor drawing, but what happens is this cell wall is actually pinched off from the inside out. So it actually like starts to build the cell wall inside the cell, and then grows, and then it will section off. It doesn't pinch off like a contractile ring. It just kind of grows, and then it's done. So that's called the cell plate. The other one is called this contractile ring. All right. So that's the ins and outs of mitosis. So if you have any questions, give me a call.